My name's Kevin Schooley. I'm a board member with the North American Strawberry Growers Association, and I'll be helping moderate this session this morning. Our first speaker is Lorena Lopez, and she's going to talk about the impact of pesticides on predatory mites. So, as we all know, mites become an issue, I think, for every strawberry grower, and any way we can better learn to manage them is a great asset to us all.
Okay, good morning, everyone. Thank you for being here, the start of this session. So I appreciate you coming, and thank you for the introduction, Kevin. So today I'm going to talk about the effects of the pesticides on predatory mites, and basically I'm going to go over 3 different studies that are out there and resources that are really useful for us to have an overview of how pesticides, a different type of pesticides, can affect predatory mites when we're using them in strawberries specifically. So I'm going to start with a brief overview of the predatory mites that we mostly use and we've seen being tested on strawberries for pest mite management and also things like thrips. And so you're probably familiar with Phytoscelis persimilis. It's one of the most largely studied predatory mites. It has been used and reared commercially for decades. And it's a very voracious predatory mite that feeds only on spider mites, meaning from the family Tetranychidae. As you can see, it can eat a large number of eggs and prey per day, which makes it a lot voracious. And it's very commonly used not only in strawberries, but other small fruits and also vegetables for the control of spider mites. Then we have generalist predatory mites, meaning that they can eat multiple types of prey between pest mites, pest insects, and most of the time pollen. We have Neosillus californicus, which is the second more commonly used in strawberries, sometimes alone, sometimes together with persimilis. And these predatory mite prefers feeding on spider mites, but can also feed on cyclamen mites that can be found in strawberries, and some occasional infestations with brood mites that are in the same family as the cyclamen mites. And sometimes can feed on pollen, but that's like the least preferred food. And it's not, as you see, it's not as voracious as the obligatory predator, but it still can be really effective in strawberries. Then we have one that has been lately evaluated in strawberries, which is Amblyssus swirskyii. This is a generalist predator that usually prefers to feed mostly on whitefly management, on whitefly pests. And well, in most areas, as far as I know, we don't have that problem with whiteflies. And strawberries, reason why this predatory mite is often more used in vegetables when we have those issues. But they can also feed on a bunch of other prey, like thrips, tarsoninid mites, like cyclamen mites or brood mites, and they can also feed on pollen. So it's a very generalist predatory mite. And I'm actually really curious because all these predatory mites, besides the preference of what they eat, they also have a preference of where they establish. So, for example, Phytoscelis persimilis and Neosillus californicus, they don't have a problem with going after the spider mites and going into that webbing that they create. Each spider mite has a different type of webbing. It could be more dense than the other ones. And Californicus and persimilis are just going to go at it and go for them. Amblyssus swirskyii, they don't like that much the webbing that pests do. They're like, oh, I have webbing, I don't want to get in there. I'm just going to go and find something else to eat. They do not have that. Most of the strains of Amblyssus swirskyii don't have that preference or ability to get in there. And it's the same on the plant host. So for example, they perform really well on plant hosts that have very low number of trichomes per leaf. So things like peppers, they love staying in peppers, they reproduce really well, they feed on the pepper pollen, but they don't like tomatoes because it has a lot of glandular trichomes that it's just kind of a stifle their movement, whereas Californicus and persimilis does. Strawberries have a lot of hair, so I've been surprised that they have been able to see some performance to go after cyclamen mites and spider mites in strawberries. And lastly, we have native predatory mite, Neocelis phallacis. This one is native of the U.S. It's also a generalist predator that feeds a lot of spider mites, different types of spider mite, not only two-spotted spider mites. And unfortunately, it has been hard to find providers that commercialize these predatory mite, because I personally think that having a native that can be introduced in some areas when we already have it naturally occurring, it's an advantage. There's still some smaller providers that have it around, and it performs really well in areas with temperate climate. Whereas Californicus, for example, is widely used in protected structures because it can withstand warmer and hotter temperatures. So this is just an introduction and a background for us to discuss multiple studies. And I'm going to start simple, and I'm going to show you a study that was done on Neocelis phallacis. And it's just going to like break down how usually these research are conducted. So we have this paper for 2016, and what they did was they went out there and they collected multiple population, well, actually one population naturally occurring of Neocelis phallacis. They came and brought the adults, they established a colony, and they tested multiple pesticides, insecticides, and miticides, and evaluated. It was sprayed directly into the predatory mice, let it dry, and then checked for a few days, 4 days. And then this is what they found. So basically in the graph, you can see that the only two that have significant non-target effects, meaning it was harmful to the predatory mice, in this case was the adults. It was, and pardon my pronunciation, Ephenvalerate, and also carbaryl. So basically, these are widely used and commonly known in strawberries. And these are from IREX 1, meaning mode of action, 1A and 3A, which are 1A, carbaryl is a carbamate, which is a very old product, and then we have pyrethroid, which is commonly known as broad-spectrum insecticides. This is consistent with a lot of other studies that have done, not only with phallacis, but with other predatory mites, that had also shown that carbaryl activates or exacerbates the population of spider mites, basically because we have these phallacis establishing naturally, and then we come and want to knock down the spider mites, but we're doing more harm to the natural enemies. If we clean out all those natural enemies, the pest is going to come back. And this time, there's no natural enemies to keep them at bay, so the populations increase even more than what we had before. So that has also been reported in citrus and other crops with this carbaryl and spider mite interactions and applications. And then when we go here, they calculated the lethal time doses, so LT50, which is basically the time at which 50% of the population that was tested, that was exposed to those pesticides, died. And then the smallest, the most toxic and faster it's going to be killed. And you can see here carbaryl and these other pesticides effectively had lower LT50s compared to the other products that were tested. The ones that had the highest LT50 was actually spinetoram. Now, we're going to dive very deep into another study that is not just focused on one predatory mite, but is actually a meta-analysis of pretty much everything, or a lot of what we can see has been tested on non-target effects on predatory mites, which are from the family Phytocidae. So this was published in 2021, and then they got from 150 populations that extracted the data of all the efficacy trials that were done with these predatory mites from the 50s to 2020, and compare and examine and mine all these effects that the different types of pesticides had on predatory mites during that time. So we're going to dive into it. And the first thing is what they found is that phytosid species not only differ by sensitivity by species, but also overall phytosid species, the adult mortality and the juvenile mortality was the highest. So we have the effect size, meaning the higher the number, the more the non-target effects, so the toxicity of overall pesticides that were evaluated, whereas fecundity and EdHatch showed lower effects of overall pesticides on all those 150 publications that were considered. And there were 3 predatory mite species that was the least sensitive species, which is a good thing that we have in that list Neosyllis californicus, because it means it can stick around even if we have some of those pesticides that we're going to dive into later. Gallindromus occidentalis is a very well known in California because it had been used for decades for in apples to control spider mites and has already a pesticide tolerance history. But I'm going to go into that later. I'm not sure what you're saying. Anyhow, in that paper, they evaluated a couple of hypotheses. So they assume the specialist phytosids like Persimilis that only feeds in spider mites that were long associated with Tetranychus species, like two-spotted spider mites or other Tetranychus species, have the opportunity to develop resistance. And then when they compare all these studies, they did supported that hypothesis. So the non-target effects were larger for the generalist species compared to the specialists like Phytosilis persimilis that only goes into spider mites. Gallindromus occidentalis is also an obligatory specialized predatory mite. And then they also evaluated one premise that was like, okay, in the International Organization of Biological Control suggested, and it has protocols to evaluate these pesticides on phytosids and other natural enemies, and they suggest and recommend to evaluate them especially on juveniles because they're assumed to be the most susceptible stage. However, that was not supported based on all the meta-analysis that they put together. It was supported that actually the adult stage was the most susceptible stage, followed by the juveniles and then DX, which is really interesting because ideally we want to test all the stages, but sometimes you only have the option to you go for the more susceptible one. Now, when they compare the types of pesticides, which ones were more toxic to this phytosid species, we have different box for adult, fecundity, juvenile, and EdHatch. And you can see the higher the value, the more toxic and harmful and more higher non-target effects are here, and you can see them all. I can hardly see what I'm doing here. You can see that insecticides and the mix of insecticides and miticides were the ones that had the higher non-target effects across all stages. The EdHatch overall was the one that showed the least effects, harmful effects by overall these pesticides. As you see, they are comparable in the lower, whereas the juvenile and the fecundity and the adult were higher. Now we have the plant growth regulators are not that toxic, but as far as I know, we don't use that often on strawberries, but for other crops, plant regulators are important in practice. Now, I'm gonna walk you through to this slide. So this is breaking down all the AREC modes on the x-axis, and then we have the effects on the y-axis. And then we have different boxes for the different stages, right? The higher the effect, the more toxic, the closer to zero, the least toxic, and if it goes below 0, it's because probably it's showing some type of tolerance already. So in the red boxes, what you're seeing is basically the groups that are more toxic to, for example, adult mortality. And there we have the IRAC1s, which are the carbamates, like the carbaryl that I mentioned. We have the group 2, the group 3s, which are the pyrethroids. And then we have the group 9, the optamine receptors. We have group 21 was highly toxic to fecundity, meaning they have some sublethal effects that even though it kills a lot of adults, but not only kills the adults, but the adults that remain are probably going to have problems with fecundity, even meaning some low egg laying or non-viable eggs. And that's when we have, for example, things like phenacequin or fempyroximate, which are often used for spider mite control. We also have, for juvenile mortality, number 1 and number 2 and number 3 are important. And here are some of the examples that I was mentioning. These are the groups that we often use in strawberries that are showing these high non-target effects on overall predatory mites. Did it change? Okay, now we're going to, I'm showing you there, the groups in green are the ones that show the least non-target effects overall, or no increase in adult mortality. We have, for example, we have group 9 and group 17 and 18, which are, the 18 are the ecdysone receptors. So things like methoxyphenoside that are molting disruptors, and sometimes we use them for lepidopterans especially, so those are not that toxic. And then we have the group 28, which is also commonly used. In the case of fecundity, all risk fecundity except for groups 18 and 25, which are, again, the ecdysone receptors. And then we have, in group 25, we have things like Nialta, which are often used also, C-flumetofen, against spider mites and strawberries. So those are very important too. And since they are not as effective, those are good options to consider. This is not moving. And here they are. Those are the examples I was talking about. Now, we're breaking it down by the miticides only. That was a group of miticides and insecticides. And this is overall phytoncid species that they considered. And then we can see here again, on the bottom part is just a list of some of the miticides that they consider in their evaluation, and the one that showed, again, adult mortality is the stage that is more susceptible based on this analysis. And groups, no, actually not groups, but the first group that we have, which is mode of action 6, were the ones that were showing the highest effect, which is consistent with what we mentioned before. And then we have the 21s like phenacequin and fempyroximate, which are knockdown products that are very, very commonly used in strawberries. And then we also have pyridabine were also very impactful, whereas things like hexythiaxox and bifenacid and C-flumetofen, which is Nialta, are the least harmful for predatory mites. Actually, I was asked by someone yesterday, like, hey, do you know if these miticide hexythiaxox, I thought it was kind of not harmful because it's killing the eggs, so probably it's not that harmful to predatory mites, right? And actually, he was right. And maybe because these specific miticides, it's labeled for juveniles and egg stages, not for adult stages. And remember, the adults are to be the more susceptible. Eggs are built to withstand very harsh conditions, which reason why probably we're finding to be the least susceptible stage. And since hexythiaxox is targeting them, that's probably why we're seeing that populations are less harmful overall. Now, when we compare the different mode of actions, it's very clear, and you probably already know that older mode of actions, like the ones that are carbamates and OPs, and number 3 is the pyrethrins and pyrethroids, you can see how high are the effects on phytoncids. In the bottom part, you're seeing the species of phytoncids they consider in these analyses. Whereas when we compare with later registered mode of actions, and these are not even that new, we already have in the ARAC classification 37 groups. By 2021, we had about 29, if I'm not correct. So these are not even that new, but even you can see the difference on the effect size, how it dropped when you compare them with these older products. So we have some new selective options that are very useful if we want to keep around the Phytocilus persimilis, not the Phytocilus persimilis, the phytocid mites in general that are happening naturally in our strawberries, but also the ones that we want to introduce and cost money to introduce to keep those spider mites at bay. Now, I want to share another type of resource. As you see, I show you traditional research, then a meta-analysis that is really showing in depth a lot of these effects. And now I'm going to show you something that it could be easily used for growers and also to understand the differences in these effects among species in strawberries. So some of the big providers of biological control agents have already apps or established pages where they put the data, the non-target effect and non-target toxicity of some products on their Phytocilus persimilis that they rear. You can easily access this. These are just 2 examples, but there are some other providers where you can access that, and it's going to give you different categories of toxicity of multiple products used in agriculture for each of the predatory mites that they commercialize. This is really useful, and when you put them together from different providers, you can do a comparison, and that's what these people did in UF. I want you to keep in mind that comparisons between predatory mites from the field and from some of the biological control providers can differ. So it is different if you go like in the first study and go and collect it in the field, that if you get some commercialized predatory mites. Reared, even if we have them reared for a few generations in the lab, they're going to get different behavior inherited into the different generations. And it's the same with the commercial strains. There are different populations that have different behaviors, and depending on the first original strain that they are rearing, that's the behavior that we're gonna see in the field when we release them. So if there's a strain that was reared that used to disperse really easily and really fast, chances are that that's what's inherited. Depending on how long it was reared, they could also lost that genetic character. Dispersal in predatory mites is determined by genetics. So based on that strain that is reared or the amount of diversity that they can put on those colonies that are reared commercially or in the lab, that's gonna differ in the field. So that's why it's important to have these and also the naturally occurring evaluations. What they did was that they compile all these different toxicities characteristics from those companies and put a very easy to follow sheet. And this is just one page. It's a longer document that is easily taken out in the field when you're planning your rotation. And you can see that it includes Persimilis and Californicus, right? And then we have, in the bottom, we have Brigade, which is bifenthrin, very commonly used in strawberries. Group 3A, the one that I mentioned, the broad-spectrum insecticides that have shown very high toxicity, as it is shown here. Also here, this is for conventional insecticides and miticides. You can see that most of them match for both species, but we have Actara, for example, is more toxic to Persimilis based on those experiments that Californicus. And then this is also useful for some of the products that we have for organic production. So They also evaluated here, but I wanted to show you in the red square. Hopefully you're seeing it. Yes. In the red square, it's just highlighting in these organic products how you can see so many differences between the two species. So you see it highly changes. Also, the MPD is also widely used in strawberries, however, is highly toxic for both. So all these tools are very, very important for us to consider. I'm going to go quickly into fungicides because I'm not a plant pathologist and I also have only 2 minutes left. And they show that basically group 6 is the one that showed the highest toxicity to predatory mites overall, with one that it's already showing in some of those predatory mite species some pesticide tolerance. Chances are those specialist predatory mites are the ones that are showing this pesticide. And then we have ones that I have been tested, and hopefully I'm going to show the results next year, is that Captan and Thriram, which are ones that we're relying a lot for NeoP, are relatively safe against predatory mites, so those are good news. Lastly, herbicides is largely lacking research, but if you're interested in going into this, there's this recently new meta-analysis that is really similar to what I show you here and showing specifically in herbicides that are showing to be a little bit more toxic than what I show you. To summarize really quick, the impacts vary by species. It varies by also the stage, and adults are the most susceptible, and depending on the modes of action, 1, 3, 6, and 21 IRACs, which are commonly used in strawberries, show the most increase in adult mortality. And unfortunately, things like fenfurosemide, that is really common, it's also very toxic, but we have some others like these ones right here that we can consider and should consider more when we have predatory mites out there. Thank you for being here, and I will happy to be around and answer some questions.
Our next speaker is Guido Schnabel from Clemson, and he's going to talk to us about a near and dear new pest that all of us, I'm sure, have heard about, NeoP. I'm not going to pronounce the whole thing, but Guido's got some good information about group 3 fungicides, because they're very, very commonly used by strawberry growers. So I'm quite interested to hear his presentation.
Thank you so much. Sorry for being late. The talk should have been uploaded this morning, but there was an onslaught on the ferry this morning. I don't know if you were part of this, but not everybody got on the ferries. Rebecca Melanson told me that she has some freebies. So there's a picture guide for strawberry diseases that she prepared. It's laminated, beautiful thing, plus some lenses, right? And you can find those picture guides over there on the table together with the lenses. So what I would like to do today is talk to you a little bit about the side effects of fungicides. In this case, Lorena talked about side effects of insecticides and fungicides on predatory mites, but in this case, I'm going to talk about side effects of fungicides on the plant itself. And so we did a few studies that I wanted to talk to you about today. One study is about switch, and the other study is about DMI fungicides. So we all are scared about Neopes. Last year was really bad for South Carolina growers. You can see here on this slide, just making sure it's the right slide. That's a scenario from last year. Same cultivar, same day of planting, different nursery, and on the left-hand side, the crop was devastated by Neopest. On the right-hand side, the same cultivar planted at the same time, also bare roots, were looking great. So no secret, there's a nursery issue. We're going to talk about this a little bit later too. This year it looks much better. So I think whatever we did last year and leading up to this year, it worked. We have very little issues this year in South Carolina, at least not that I could see, on cutoffs around the state and on plug plants around the state. But if we do see lesions that look suspicious, like the one on the right-hand side, you do need to monitor and make sure that we are applying the right fungicides to control whatever disease that might be. In this case, it could have been neopest, pneumonia leaf blotch, but it turned out to be none of those two. It turned out to be something very different and not something to be worried about. I always like to show this slide when I talk about initiating strawberry plants in the season, because I think dips over the past have been very favorable. We used a lot of switch in dips, for example, and this is a slide that coming out of Florida. I like to show this one because it has some really nice information. On the bottom, there's that non-treated control, and as you can see on the right-hand side, the disease incidence was 16%. So if you didn't do anything, you would have 16% disease. But switch, for example, basically cleaned up the disease, and you ended up with only 1% disease, which is great. Zivion is another product that I'm personally also trying to promote because it is one of those compounds that works against botrytis and anthracnose. I don't think it has a lot of efficacy against neopest, unfortunately, but it is certainly something we need to take a look at. In this particular study, Topsin M was also effective, so I think maybe that study was done on the GLOW, Calototricum glowsporides, which is susceptible to Topsin M. But Zivion looked really good, and Zivion, the active ingredient is natamycin. If you're not familiar with natamycin or Zivion, it is marketed by DSM, and the contact information is John Farragher. Hopefully he's still with the company, but I think last year I was in touch with him, and he said that they're distributing in the United States, but I have talked to several of you, several growers, and it doesn't seem to be of widespread use. That's another study out of California showing that both Zivion and Switch worked really well. You can see, according to those bars, in the untreated control, you had a lot of disease. Big bar on the very top. On the bottom, you have very little disease, small bar. Mirus Prime, Switch, Zivion, they all look very good. So let's see what. So let's talk about stunting. So there's some reports out of Florida, and we've got Marcus here. He can maybe confirm this or disconfirm it later in the discussion, but there's some reports that I heard from Florida that switch caused some stunting in some trials. And so that's something that needs to be investigated. And I think they're actually looking at alternatives, like applying switch to the planting hole rather than applying switch to the plant itself as a means to not have this stunting going on. But, you know, we've used switch for years in dips, and I've never really heard of any stunting. So unless they've changed the formulation, I was just not sure what to make out of it. And so we did a trial, and I want to thank Andy Rollins for helping me with this trial. I don't know if he's in the audience, Andy? No? Okay. Well, I did this in collaboration with a farmer in South Carolina, so thank you so much for working with me on this one. And I think it was an interesting study, although it wasn't the perfect study, but we learned some stuff. The study was done on Ruby June and Fronteris. The Ruby June plugs and plugs, in both cases were plugs, Ruby June and Fronteris plugs. The Ruby Junes looked weaker than the Fronteris, and keep that in mind because I think this is something important to remember for the results, interpretation of the results. So we looked at switch and we looked at Zivion in dips and in drenches, but we left the plants in the plastic trays for 2 weeks before planting. So that's not according to label instructions, but at least the plants weren't like constantly exposed in plastic bags like you would do with bare roots or cutoffs. Instead, they were left in the trays and they were irrigated, so they were watered and stuff. It was a replicated trial. So those are the results. Again, Ruby June on the one hand and Fronteras on the other hand. Plant health was measured on a scale of 0 to 3 for Ruby June and a 0 to 4 in Fronteras. So for some reason, we used different methods. And we looked at Zivion dip, Zivion drench, switch dip, and switch drench. And we found that in the fall, when we rated the plants in November, there was certainly an impact that we observed with the switch dip and the switch drench. And as you can see, the control had 2.3, so the plants look pretty good. Zivion dip and drench 2.1 and 2.3. I'm looking at the fall column. And switch really reduced plant growth in a way that we could measurably demonstrate that and see that. The effect lingered on to the spring. Again, the switch plants were much smaller than the control plants. Now, what was also interesting was the plants that were treated with Zivion actually showed a positive response. So if you look at Ruby June spring, control is 2.1. The plants that were treated with Zivion were 2.4 and 2.6, so they were healthier, better looking than the control plants. On the other hand, the Fronteras plugs, which looked much better at the beginning of the experiment, they didn't show any signs of stunting in the fall and no signs of stunting in the spring. So in conclusion, what that all tells us is that switch can stunt plants and the effect will carry over into the spring, but the stunting effect may be dependent on the plant quality. If you already have plants that suffer from something or that are just in general weak, I think those plants might be more susceptible to switch toxicity. On the other hand, the plants that looked good and were strong, those plants, there was no negative effect of switch on those plants. I don't know if you've observed similar things in Florida, but we can talk about this later. So to me, there was no solid evidence that switch dips actually conducted on normal, strong plants cause any stunting. But what was also interesting was that in general, the weaker Ruby June plants benefited from the Zivion dip. So there was certainly some positive element to it. Now let's switch gears and talk about DMI fungicides. DMI fungicides have gained a little bit more interest among farmers because they are effective against neopest disease, especially if we mix them with Thiram or with Captan. There seems to be a good effect on neopests. So it's in our best interest to learn more about those. There's 3 different FRAC 3s out there that we can use: propiconazole, flutriafol, and difenoconazole. The trade names are Tilt, Propistar, and Bumper for propiconazole, Rhyme for flutriafol, and Inspire for difenoconazole. Now, propiconazole, ask any nursery, is something that can stunt plant. It's a growth regulator. And it's not sold as a growth regulator, but it acts as such, especially on small plants. So nurseries usually stay away from propiconazole. And there are a couple of studies I found they were conducted also in Florida, where there seems to be evidence that propiconazole, if applied on a regular basis, multiple times, that there's some stunting. So we looked at this at the Musser Fruit Research Farm. That study was supported by the South Carolina Small Fruit Association. Thank you, Bob Hall, for supporting the study. He was instrumental in getting this done. We looked at Propistar, we looked at Inspire, and we looked at Rhyme. And we looked at them in the fall, so we applied those either one time in the fall, 10 days after planting, or we applied them 3 times in the fall in 10-day intervals. So that's what this means over here. The number of applications, 1, 2, 3, means 3 times they were applied, and the number of applications, 1, means it was only applied 1 time. So this is what it looked like. This is our spray rig and our beds that we formed, and at later in the season, this is what the block looked like, and you can sort of already see that there was no detrimental effect of applying propiconazole, for example, 3 times in a row. Those plants look pretty good. What we did was we measured the diameter of the canopy and the height of the canopy to assess plant health. Those are the results. Again, you've got your treatments, Propistar, Inspire, and Rhyme, applied either once or 3 times in the fall. Each treatment also received or entailed Thyram. So Thyram was always added to the spray tank because we always mix those things. We saw then that there was very little difference between treatments. So I don't know, can you see can you see that cursor? No. Okay. So where it says canopy diameter 22 of November, those numbers all look pretty much the same. There was a little bit of difference in the treatment where Propistar was applied 3 times in a row, but really, I mean, we're talking about 5 to 10% plant growth reduction. When we did another assay, another evaluation in April, and the same thing here. There was a little bit of something in the Propistar treatment, but not that much. And then we also looked at yield. There was no difference in yield that we could see. So yes, if you apply propiconazole 3 times in a row in the fall when the plants are young, you will see some stunting, but I don't think it's a big deal. But still, nobody wants their plants to be stunted. So keep that in mind as we move on to the next slide. This is the spray guide that I'm proposing. I've done this in several sessions at several meetings. We can talk about Thyram because Thyram seems to be in shortage this year, but Thyram there's 2 spray scenarios, A and B. The first one is when you have healthy plants and you don't really have any evidence that there's any anthracnose issues or any neopest issues. Whether or not you're going to apply Thyram in the fall is up to you. Maybe before you apply the row covers, might be a good idea to apply something, but you don't really have to. Then as you move into the next year, your backbone should be Thyram and Captan, and you would add something more effective only if the weather changes and you're expecting a major infection event, like right before a major rain event. So for Thyram, we would recommend adding either Rhyme or Inspire. Stay away from propiconazole is my recommendation because there is a little bit of stunting to be expected, and nobody wants any stunting, regardless of there's a yield. It might not be a yield detriment. But if you want to use it once or so, that's also fine. When you use Captan, we recommend that you add Inspire Super or switch to Captan as you move into the production season. But make sure you don't use, especially switch, more than 4 times if you use it at the full rate or 5 times if you use it at the low rate. Still good in time. Still some I want to make sure that you know about the resources that we have for our growers. The Southern Region Small Fruit Consortium has excellent resources at www.smallfruits.org. So make sure you are aware of those, including the spray guide. Make sure you have downloaded the MyIPM app. We update it frequently. We have 2 new specialists who are very eager to give you the best information, not only in the spray guide, but also in the MyIPM app for strawberries and for blackberries and for blueberries and so on. So make sure you have that one handy. And I do want to acknowledge all the people that were instrumental for generating all this data. Johanna Wäsche helped a lot with the field trials for investigating DMI fungicides. Andy Rollins was instrumental in conducting the switch trial. Julia Gillane, my lab manager, Master Fruit Research Center, and of course Bob Hall and the South Carolina Small Fruit Association. So thank you very much for supporting our studies and our efforts. Bob.
Peter, was to repeat us the power I mixed with propanol in the study. I couldn't remember, was that previous year?
So we did some studies where we, so the question was, if we mix howler with propiconazole, would that be of benefit? Is that the question? So the answer is yes. I think there is some synergy there. We tried to reduce the rate at which Propistar is being used, because if you reduce the rate, you're also selecting less for resistance, especially if your control success is not reduced. And so that was our goal, and we showed that if you combine the full rate of howler with a reduced rate of propiconazole, you will get the same efficacy as if you applied propiconazole at the full rate, at the high rate. So that was our purpose. But if you combine the two at the full rate, I would think that you also have an additional effect.
Are there any other questions? You know, we have time for.
I would be interested to hear from Marcus about the switch experience in Florida. Marcus, do you want to comment on that one?
Yeah, we see more like the stunting whenever we do the tipping, or whenever we do the foliar spray, then we don't see the.
Can you stand up and talk to?
Yeah, we see more the stunting whenever you do like deep treatment, but whenever you do foliar spray, the switch, they're not
So you do see stunting on a regular basis with switch dips. Is that what you're saying?
with dips, yes.
With dips.
Foliar spray.
Foliar spray, no. But dips you see even on strong plants. On plants that are not, okay. So that's different from what I experienced. But I use plug plants from my experience, from in my experiment. So I haven't done my studies on maybe bare roots or, cutoffs.
Do you have any thoughts on whether that stunting might be the switch affecting the plant physiologically or effects on the rhizosphere of the plant?
So I haven't looked at the why yet, but I would think it's probably a direct effect on the plant. That is my guess, that there's some sort of physiological effect that makes the plant get in some sort of a shock, and then it takes a while to get out of it. So even if you're not going to not have any crop, right? So it's just that everything is a little delayed. Is that fair to say? Yeah.
One more question?
Can you clarify dip versus a drench?
Yeah, so a dip would be you dip the whole plant in the solution for several seconds, and then you plant them. A drench would be, you would do this on the trays. You would drench your trays with a certain volume of your fungicide, and then it would then go through the roots and the soil and then drain. That would be a drench.
I have a quick one, if we don't mind. Who's looking at fungicide resistance in particular my concern with the switch, and even last fall when we were fighting the infections so much, and you guys and Bart, we were working on spray rotations, we're talking about fall applications of switch. We don't know what's been done in the nursery, right? So how much efficacy? My concern is we hammer this stuff in the fall, then we come into the spring and we've got botrytis outbreaks, and we've used up the real efficacy. Who's studying it? Who's looking at it from a long-term perspective?
I would say Florida is studying it, and we are studying it as well. We have a plant problem clinic at Clemson University. They are using our method that we developed in my lab to inform growers about the susceptibility of the pathogen to various fungicides. Fludioxonil in switch is part of this. So we are monitoring, and we haven't really seen a whole lot of resistance coming through the nursery yet. I think it's different in Florida. In Florida, they're seeing a little bit more resistance creeping up, so maybe at a little bit higher level. But it's a great point, and I'm trying to pound that information that we should not overuse switch, because it is prone to resistance development, and if we lose switch, we're in big trouble.
Hey, if I could add something to that, don't have anything to quantify at this point, but and I ought to keep my mouth shut because it's a trade business. But I haven't used a lot of switch in my nursery, and I think I could point to some earlier susceptibilities to botrytis in the fruiting field in Florida on berries, and that is interesting to me. See, because I think that I am managing resistance so that they can use switch when necessary. But and it's not every variety; it's a couple of specific varieties we've seen maybe a higher susceptibility to botrytis in some of my stuff than not. So.
You can make this a sales pitch. Nurseries can make this a sales pitch. We are not using switch. We are not pre-selecting for resistance. So if there's a very good chance that botrytis that is definitely going to come in with the transplants, there's no way around it, has a very good chance of being susceptible to the fungicides you need and you will use. I think it's a great sales pitch. Thank you.
All right, before you go, Guido, I'd like to thank you with a gift from the Southeast Regional. Thank you for speaking.
Thank you.
You're welcome. All right, and I forgot to give one to Lorena, but I do have one for you here as well. Yes, which one is yours? This one?
This one.
Okay. I'll tell you in a second. Okay, just a reminder that all the sessions are being recorded. Thank you, Guido. All the sessions are being recorded, so you'll have an opportunity to see them at a later time. For our next talk, we have, I guess, a panel or a discussion between a couple of propagators. I think Kyle and Nora. And Kyle, you have a presentation. Nora, do you have a presentation or just gonna? No, do you have something on a PowerPoint or anything, or are you just speaking? Okay. All right, I'm going to load this up. If you want to come up, Kyle, and so they're going to talk about NEOP again, and and how they're working to reduce the potential of NEOP incidents in nursery plants. So it's a great topic. I'm sure that's of interest to everyone. As we know, there's been some tough years depending on which area of the country you're in with this new and challenging pathogen.
Hi, everyone. Just for those of you that don't know me, I'm Kyle Dick. I'm the crop care specialist at Easy Grow Farms in Langton, Ontario. We're a strawberry plug and tip producer in Ontario, and essentially talking today about some practical management techniques of Neopestalotiopsis in nursery. Please just keep in mind I'm not a scientist, but I hope some of my definitions and terms are practical and we can use them moving forward. So a little bit about Neo, just from how I believe it to be so opportunistic and so hard to predict, is it's an opportunistic fungal genus distinct from Pestalottioptis and characterized by uniquely colored spore segments, as you can see in the pictures to the right. It produces dense asexual spores that are typically 4 septate, meaning it has 4 cell walls, and you can kind of see the cell walls in the picture to the right. And the reason I use the word dense is because I refer to it as a really thick-celled wall pathogen, meaning that it can be very difficult to eliminate because of how thickly cell-walled it is. As most of us know, multiple species and strains exist in North America, and some being more pathogenic or more aggressive than the others. Wide range of common host plants, including agricultural and ornamental plants. And again, a lot of you know it can be oak trees, rhododendron species, guava, mango, blackberry, blueberry, strawberry, of course. Neo can act as endophytes, meaning that it can live within the vascular tissue of plants for a long time before actually seeing symptoms or ever seeing symptoms. It can also act as a saprophyte, meaning it will sporulate and develop on decaying matter in your field, and also just as a pathogen. It's widely identified across North America, as all of us know, and it's frequently found as a disease complex, most commonly with Phytophthora species and Calototricum or Anthracnose species. So what I'll be focusing on today are influential factors in our mind of managing Neo-P and strawberry propagation and in nurseries, and talk about clean mother material, growing techniques, irrigation, water source and management. Two of the big ones would be nutrition and IPM techniques, a little bit on spray management, cleaning and sanitation and testing protocols. And then I think something that's a big factor is just putting all of these things together. And without a couple of them, we can still run into big issues. So from a mother material standpoint, I think we all know it's crucial for success all the way down the line. To start out with, ensuring that the timing of your mother planting, both in the field and in the greenhouse, is accurate. The last thing we want as tip and plug producers is to run into a time frame where our tips are starting to age and they're starting to show symptoms, and in return we're more susceptible to diseases in general. Test and scout your mother material. This can be both at the nursery you're getting your mother material from, on arrival, cutting crowns open, and just overall evaluating what you're seeing is really important in our mind. And if you see something that's concerning, having your team address it with you and your team, it can be pretty important. Plant sauna is something that we purchased in 2024, and it's a heat steam treatment. By no means is it the end-all be-all to Neo management, but it does help, and we do use it on most of our mother material. Diversifying your mother material, so this can be, if you're planting bare root plants from California, this can be having multiple nurseries, maybe in the greenhouse side of things, incorporating some tissue culture plants and/or interline management. So it's one thing that we are using all three things at our farm, but one thing that we are doing is we fly over some BM2 or BM3 material from Europe, and then essentially strike those tips later in our season, and then put them in our cold store so that we can have all three different types of mother plants on our facility, and just overall try and reduce risk by diversifying. And then, at the end of the day, we're all farming, so be prepared to shift timings and varieties when things do come up, and really know what you want to receive. And that, I mean, more so on a root mass, crown thickness, and diameter standpoint, and overall leaf area and number of leaves. So for growing techniques, I would say, this is going to be a recap from a lot of presentations that we've all seen on Neo, so I'll try and keep it brief. But, select varieties that have field tolerance to Neo and place them accordingly. And this is more so in the field. You understand your farm and your field. If you have a low spot and you have to plant into it, potentially putting your strongest field tolerant variety in that area can be helpful. More on the tray plant and plug side of things is selecting a media that has sufficient drainage. So, at our farm, we're using mainly core-based products that have good drainage. But if you start using a combination of mixes, so let's say it's a peat and perlite or core mixed together, eventually someone has to mix that, or you have to mix it yourself, and there will be more inconsistencies, and it makes irrigation management in your farm more difficult, and from one plant to another, even in that same tray. Of course, avoid favorable conditions of spore development. Limit abiotic stress factors as much as you can, and that can include drought, salinity, extreme temperatures. maximize airflow. So in the field, of course, it's headland management, plant spacing management, but in the greenhouse side of things and covered production, you know, a lot of the greenhouses we have horizontal airflow fans or vertical airflow fans, but we're not necessarily utilizing them properly. So ensuring that you are and proper venting techniques can go a long way. I think airflow is something that really needs to be maximized, especially in summer production, because it's stressful on them. Remove nearby host plants prior to planting. Proper field prep, and Dr. Stanley's presentation yesterday on fumigation I thought was really good, and just overall being prepared in your field. And then of course manage labor according to disease pressure, and that can be minimized plant handling when things are wet or moist or early in the day, or just overall, if you run into a disease issue and you start removing plants from your issue, really making sure that those hands aren't touching some of your healthy plants. From an irrigation water source and management, I'd say this is a pretty big factor, and one of the biggest things is, what are you starting with? Each well system, lake, pond, stream, they're all different, and understanding what elements are in there, the hardness, the pH, the amount of iron, they can all play a role into into what you're finally feeding your crop and your targets. Understand how clean your water is. So, a lot of the university programs, a lot of the accredited laboratories in Canada, the US, the Netherlands, they have quite a bit of technology to help you understand how clean your water is, and ensure that that's not part of your problem before you even get started at your farm. And with plating and culturing, some DNA sequencing, they can tell you if your water is part of your problem or not. Properly disinfect your water if necessary. And so if you find out that your water source is part of the issue, or if you're in a greenhouse and you're forced to reuse water, some of the things that we found to be the best at either removing or damaging and killing these spores are fine filtration, and that can be, ultrafiltration, where it's a really small micron filter. It can be cloth or paper filtration. Even just a disc filter coming out of your pond can go a long ways. We do use ozone disinfection in one of our greenhouses, and we do find it to be pretty handy. It also adds some dissolved oxygen to your water, which in the summertime can be a pretty big benefit. And then there's some oxidizers, of course, the hydrogen peroxides, the hypochlorous acids. Some of these can be your final inline treatment to not only keeping your irrigation lines clean, but potentially killing and damaging some of these spores. Create backup solutions if you can. This can be anything: having an extra well, having a nearby stream or pond that you can pull from. Tailor your nutrition program according to your water tests. So we all have our nutrition targets, and we all know what they are, or at least have a good idea of what they are. But again, if we're not comparing them to what we're starting with in our water, then our targets aren't really our targets. So, really comparing these things together can be really important. And then tracking the amount of dissolved oxygen in your water. You can buy a DO meter on Amazon for like $20. Keeping an eye out throughout the summer just to see how much dissolved oxygen, it can go a long ways in keeping really good and healthy root health. And overall, I would say a baseline that we're looking for is, if dissolved oxygen levels in our feed water, both either in a field or in our greenhouse, if it gets below 7 or 8 parts per million of dissolved oxygen, I'd start to get concerned, and I'm going to find a way to add some of that dissolved oxygen back in. And there's some pretty cheap methods of doing that. From a nutrition standpoint, I do think this is one of the biggest factors in managing neop in the greenhouse. Have a nutrition plan prior to planting. Get recommendations from your consultants, from your breeders, that they have that information. Quantify total nitrogen as best as you can. This can be, not only from an economic standpoint in your field yearly, but also just if you're growing plugs or tray production, quantifying the pounds of nitrogen per acre per week based on a certain age of tray plant. I think it can go a long way. And then selecting the proper form of nitrogen. So we're using single element, we're using some bag product, some premixed bag product, and really understanding what form of nitrogen it's in. I would say the safer way to go, and it will be a little slower, would be sticking with nitrate nitrogen over some of the ammoniacal and urea sources that, the ammoniacal nitrogen source, it can acidify things in the root zone. There can be a benefit to that, and it can help speed things along. But if you lean towards more on nitrate, I believe that you have thicker crowns and more stronger, compact plants, especially in your stressful times of year. Use plant sap analysis and dry matter testing to monitor uptake and balance. And plant sap analysis, there's some labs in the U.S. that does it, but it's giving you a real-time shot of what's in your leaves in the morning, and it can be a little quicker than dry matter testing, where you might be a week or two behind of what's actually going on within the structure of your leaves and in your plants. Monitor sodium and chloride levels in your plants. When sodium and chloride levels get too high, they can start to become relatively toxic to plants, and just having an eye on them. Ensure source water aligns with your targets. So just comparing those two can go quite a long ways. And then use element ratios that help produce the most compact and strong plants you can, and that can be using a fertilizer program that has the lowest amount of nitrogen in total, and you have really high microelements and high calcium and potassium levels, but you're still able to feed every single day. As for IPM techniques, I think diligent scouting goes a long way. A lot of us are incorporating beneficial insects on our farms because of resistance management and whatnot, and a lot of the companies that sell these beneficial insects, they've got a really cost-effective digital platform, or free, for instance, and for year-over-year data collection, especially as your farm gets bigger, it can be really handy. Fungicide management, which I'll talk a little more about. Incorporating some biofungicides or biobacterias just to inoculate open space. And again, Howler Pro came up earlier. That's a great bacteria-based product that can, relatively cost-effective and can go a long ways in just taking up some of that open space before something else does in the media. Incorporate compounds that help support protein and enzyme production in your plants, and these can be amino acids or organic acids, and even in a small way, some humic and fulvic acids. Use a preventative spray model for Neo and set thresholds to when it's time to take out your stronger bullets. Calibrate your equipment for different types of applications. We refer to spraying, drenching both inline and over the crowns, and then what I call sprenching. So essentially somewhere in the middle between spraying and drenching, and getting the active ingredients closer to the crown, specifically those systemic ones. As for fungicide spray management, I won't go too far into the weeds because we just had a great presentation on it. But sometimes you got to use larger droplet size and increase your spray volumes when canopy penetration is needed. You can use bacteria-based products in between your synthetic fungicide apps and not have to worry about damaging them as if you were to use with maybe a trichoderma or a fungi-based product. Avoid tank mixes with the same active ingredients of the same mode of action. Again, so those multi-site fungicides being mixed with a group 3, for instance. And then keep fludioxonil or switch for your timely matters. And that's when Neo is present, high stress periods. If possible, if you're building your own fungicide plan prior to the planting season, I would build it around your multi-site contact fungicides, and that's going to be Thiram, Captan. We've got Bravo in Canada, and then where does copper fit in? I don't think there's too much efficacy with copper-based products on Neo, but I still kind of refer to it as a multi-site product. Of course, rotate which single-site products are used and rotate between multi-site and single-site when you're not mixing them together. And continue your preventative applications for Phytophthora and for Pythium, Anthracnose and whatnot. Again, that just goes back to Neo is found as being part of a disease complex in some studies. And then, having open communication with growers and nurseries. I want to make sure that we're doing everything we can to keep your plants clean, but at the same time, if I use switch right before I sent the plants, I wouldn't want you to see you guys use switch right when you plant them. Those types of things, and just being really open can go a long way. As for cleaning and sanitation, I would say the biggest thing that's often missed is that we're not necessarily cleaning before we disinfect. And so if we don't remove the pathogens home, that can be, a grease on a gutter or, dirt on lay flat. If we're not removing the pathogens home, the disinfectant's never going to really work. So you try and use alkaline or soap-based products for removing your oils, greases, and your dirts, and acidic-based products for removing your salts and calcium buildup. And then disinfect with products that contain multiple ingredients. The quaternary ammonium products are great. They've got a residual. They might not be the strongest against some of these pathogens, including Neo, but there's products out there like Multides or Virocid that have multiple quaternary ammonium products, also have isopropyl alcohol and glutaraldehyde, and it's all in one product. It's pretty safe to use, and you're kind of getting a 4-in-1 shot. Focus on touch points the best you can. Try and think of everything that comes in contact with infected material, from tools, equipment, vehicles, shoes, and then have biosecurity measures in place to prevent bringing something into your farm. As for testing protocols, I'll leave this to the universities and to the laboratories, but, visually inspect plants often, even non-symptomatic plants, cut crowns open, and just look for issues all the time. Create a testing SOP and follow it at the beginning of the season. If you don't, if you put it in your calendar that you're gonna do something every 2 weeks or every 3 weeks, if you stay true to it, it can help you in the long run, especially with some of the more advanced testing techniques using metagenomic sequencing or some sort of DNA sequencing. It can give you a real-time shot of what's in your water, what's in your drain samples, and overall help. This year we did a little bit of heat mapping where we were monitoring some of the slab and drain water and just water near our root system, and just keeping an eye on where the levels are, and it helped us. Plating and culturing your samples are really important, understanding if it's really alive or not, and then use a trusted laboratory that you have. But yeah, this is a little bit about us, and I think Nora, you want to come up and we'll do like an open type of panel for.
Thank you, Kyle. Next up, I'm going to invite Nora Dorgan from West Tech Greenhouses in Prince Edward Island, Canada. Nora's going to give a brief presentation as well, and then we'll bring both Kyle and Nora up together, and you can ask questions at that point.
Okay. Good morning, everyone, and thanks for coming and showing your interest. My name is Nora Dorgan, and I'm from Prince Edward Island, and we have a strawberry plant nursery called West Tech Agriculture. We were, unlike many other nurseries before us, that had a NEOP problem last year, and it was our first eye-opener to NEOP. Okay. We started to say, what happened? Okay, so first we're trying to figure out what happened that we had a NEOP infection, and then after we realized what we think happened, we had to move forward on how to deal with this. So just a little bit of history of what we think happened. We produce our own plants. We have an internal nursery, and a couple of years back, there was a problem sourcing the Ruby June variety. So the master licensee just mentioned to us, we'll just hold over more plants and keep them an extra year. Having not known that we possibly could have had some NEOP in 2023, a small amount of inoculum, holding over plants is not a good idea, but we didn't know we had small incidents of NEOP. Otherwise, that would have been gone and new stock would have came in, and it's all about keeping the inoculum low. So holding over those plants, and then what we did, we put row cover because people wanted the plants earlier. So putting row cover over plant material that possibly could be infected with NEOP probably increased our incidence of NEOP in 2024. So having realized we had NEOP problem, what did we do? Well, first, I reached out to as many specialists as I could think of, and The first one that came to mind was Dr. Natalia Perez from the University of Florida, and I asked her for guidance. Her biggest, she said, reduce the inoculum. That's your big step going forward, is keep that inoculum low. And I said, well, how do we do that? She said, well, the best way to get rid of the plants. I said, okay. So that's what we did. We harvested every plant into the field and trucked them, put them in dump trucks and trucked them a mile away and buried them. So that was our first way of reducing inoculum. This took quite a while to do, but we thought in the long term it was a way of coming back. So she also mentioned that the mother plant has a big crown, and when it, after it's put into the field, that crown takes a long time to disintegrate. And she said in Florida, they did studies where after 18 months, they were still seeing viable NEOP spores in that crown material, even though it looked dead, and it is dead. The plant's not viable, but the NEOP spores could still be viable. So having said that, she didn't know in our area how long it would take for those NEOP spores not to be viable with our winters. And then I mentioned to her that we're on a 3-year rotation. She said, well, that's great. That's another plus. And, currently we're looking at going to a 4-year rotation, which will add to, if there is any NEOP left in any small crowns. She said not to worry about small crowns. It's the big mother plant crown that you want to discard, because the smaller crowns will break down in a lot shorter time. So with that advice, we moved forward, and the total crop, like I say, was trucked away. So that was our first step. Our second step was to look at our whole production system and how could we change or how could we better it. Although we produce our own stock material, we would often have that stock material in the same field as our saleable stock. So what we did is we started plans to change that, and we had another property that had never seen, hadn't been really farmed for at least 20 years. We brought that into production and we created our nursery amongst the nursery. So that's totally isolated from our production area. It's a self-contained nursery with its own greenhouse, dedicated tractors, sprayers, planters, even the garden hose. So all workers who go to this nursery must change footwear, gloves, everything prior to entering any of the fields. Foot baths became a very common piece of equipment, I guess, around our farm, and it was all about reducing inoculum and keeping inoculum away. Another immediate initiative that we started was increasing our sanitation process. All the machinery that is now being sanitized going from one field to another. If one field is just 2 miles down the road, it comes in and gets sanitized before it goes to the next field. Along with all the equipment being sanitized, we have foot baths, we supply disposable gloves and booties, and even in a field, if there are 2 varieties in the same field, we have the workers change gloves and booties prior to going to the next variety. So it's all about trying to reduce inoculum in the area. So prior to 2024, we had no idea that we had NEO. And, a lot of the people saying, if you have your own stock material, you're not going to get NEO. However, in my search for information, I'm finding that NEOP is everywhere. Like pretty well every growing area of strawberries will have NEOP, whether that's Australia, the Netherlands, Spain, United States, Canada, there's NEOP. So prior to this year, I guess, we were never using the chemistries for NEOP. And so we started to use chemistries for the NEOP. And I think that made a big difference. So you have your DMIs, your Bravos. Down here it's called Omega. In Canada it's called Allegro. These are all have some efficacy on NEOP. And introducing them to our operation, I think, has also made a big difference. Another thing in my search, they're saying that NEOP can be transmitted through wind and rain. So we have had using overhead irrigation in our operation. So what we have done is we've went from 40% irrigation and 60% drip to 100% drip. So that eliminated any kind of water dispersal. And I think that in itself would help decrease leaf wetness and help with the NEOP problem. In the past, we've also looked at the size of tips. When we send tips down into the U.S., we've always had some feedback on tips and uniformity. So after much feedback and more knowledge on what practices are acceptable, we narrowed our size profile. A more uniform size tip, we hear over and over again, helps in many ways with more uniform watering, as well as the taller tips are not draping over other cells in the trays, which can lead to more fungal problems. So that's just one small thing that we can do, keeping a uniform size tip that may help in the tray production down here. Okay. After our initial review and changes to our practices, things that we could identify as good agriculture practices, we didn't stop there. We reached out to all the people that I could talk to, and I was introduced to a lady from the Netherlands. Her name is Dr. Petra van der Goes, and she's an independent plant health pathologist. And she said that NEOP has been around in the Netherlands since about 2013. And they have an extensive strawberry industry over there. So I said, okay, well, what can you do to help us? So after much discussion with Dr. Petra, I convinced her to travel to our farm, walk our fields, and do a risk analysis for 2025. She's also has taught us methods of doing single plating to identify what might be present, in any of our plant material. And she explained a total plant collapse can be caused by many different pathogens, such as Coleoptera, Phytophthora, Macrophamina, Fusarium, NEOP. And her work is centered on the ideas that plant health cannot be improved by treating symptoms alone, especially when complex disease symptoms are involved. In the case such as NEOP, the challenge is rarely a simple pathogen. The disease often appears alongside other infections and stress factors. From her experience, she is cautious when dealing with NEOP because the problem is defined too narrowly. The solution, if it is defined too narrowly, the solution will also be incomplete. Correct identification of all contributing factors is therefore essential before any intervention is chosen. So to commence NEOP sprays after initial NEOP diagnosis must be reevaluated when a complete diagnosis is available after about 7 to 10 days when you get your full diagnosis. And if there is a Phytophthora detection and this information is ignored because all focus is on NEOP, then NEOP sprays are not going to address the underlying issues. The chemistries for Phytophthora control are very different than for NEOP control. One must be careful and be very thorough. So I feel the industry is very focused on NEOP, and some of these underlying very important issues can be ignored. As Petra has pointed out to me, that NEOP often appears alongside other infections. And is it possible that this is where it's getting its aggressiveness because of the synergy with another infectious pathogen? So I remember talking to another nurseryman who had a NEOP problem, and his comment was, the more I sprayed, it felt like I was doing more harm than good. So I shared that comment with Dr. Petra, and she was not surprised. She explained that although the sprays may suppress symptoms temporarily, they often increase plant stress, disturb biological imbalance, and ultimately weaken plant health. Overspraying can create more problems than they solve. Overuse of registered chemistries can lead to resistance, and then what? So in 2025, we encourage growers to use Aliette and as a spray for Phytophthora, and also the product Arondis Gold or Iridomil as a drip treatment. And the reason for this recommendation was also, amongst all the hype of NEOP in 2024, a farmer shared this protocol, and even after a wet spring of 2025, had to spray a desiccant on his plants at the end of the season to kill them. So, what's the future look like? Well, amongst all the interventions we have implemented, we are also adopting a program to produce more resilient plants. Plants that can be stronger in the face of adversity, that can maintain their health. We have explored the use of biological applications to our production fields. This is a totally new science for me, and the timing of applications is critical. There is so much to this approach, and if we can establish a good working protocol, it will complement our spray protocol while not being solely reliant on chemical sprays for control. With the continuous loss of chemistries, such as Thyram, that is people are questioning at this conference if it's available and if it's going to continue to be available, this approach may become more important in the future, so we still have time to be proactive rather than postponing and be reactive. From what we have seen this year, we are encouraged to continue this approach, tweaking along the way. I'd like to share another story from a plant pathologist who decided to test plants that were looking good. To his surprise, testing found positive NEOP. However, the NEO did not define how the crop turned out. Why? Perhaps the inoculum level was low. The resilience of the overall plants might have been good. And maybe there was reduced stress from other pathogens not present. So there's a lot of things at play when you have a NEOP diagnosis, and we need not to panic. We need to have an overall view of what's going on with that field. So to conclude, I would like to say that we have adopted, changed, and added many approaches, but I cannot single-handedly say which one made the biggest difference. Perhaps the solutions to a complex disease such as NEOP have to be as complex as the disease itself. That's about it. Any questions?
All right, we'll invite Kyle back up, and we can, I'll try to moderate questions as best I can.
Just take this off here. You guys, if you want to sit, you can, or you can stand, whatever's easier. Okay, any questions from the floor to either one of our panelists? Okay. Okay, go ahead.
Variety tolerance is one of the things, are you looking at that or having plant breeders look at it? Because there's some varieties that got tolerance, more tolerance to diseases, and I think that makes them more tolerant to the NEOP.
So the question of was whether breeders or others were looking at variety tolerances. As we know that there is some varieties that stand up a little bit stronger to certain diseases. So Kyle, you're the closest. I'll pass to you first.
Sure. Yeah, I'd say from our standpoint, we're definitely looking at which varieties are more, I would refer to it as field tolerant than others. I think, what was brought up yesterday a bit is we do need some of the genetics to be bred in, and where at least one parent, if not both parents, have true resistance, and that's going to take some time. But I would say that we're definitely keeping stats on what we believe to be the most strong against, and talking with everyone from the industry from our standpoint, and we're encouraging. We grow an Everbear variety in our greenhouse that seems to be pretty strong against it, and we definitely talk about that when we're talking with some of our customers.
There's definitely variety. Certain varieties are more sensitive to NeoP, and we do have, we've incorporated some new varieties that have more tolerance. But it's the industry that drives the demand. So until those varieties are tested in the geographical area where they're going to be grown, it's hard to promote them. But that's what we're working on, is trying to get them out there and get samples and trials and see if they do work in this geographical area.
Any other questions? Yes, go ahead.
What about public access to the people that do like UPIC operations as far as the transmittal of fungi in the field? How do you have your customers wash their feet and they pick the Joesbury farm and they come to Dillsbury farm? You see the disease protocol transmission with, say, hundreds of people coming to the field that have been elsewhere.
Yeah, so the question was about public access, especially in pick your own operations with people wandering perhaps from farm to farm or what have you. So I don't know if you have any comments on that.
I personally don't have too much experience with that, but it is a really good question. And I think we all understand that mechanical transmission is happening. It is important. I don't believe it to be the biggest spreading factor, but from a UPIC standpoint, I think, really trying to avoid picking your areas that are the most wet, like where the leaves potentially are staying wet the longest, is where you're going to run into big issues. If your plants are dry, I believe that your mechanical transmission is going to be way less. My only other thought would be using some of those oxidizers as sprays at the end of the day. When you're done, when the UPIC's done for the day, there are some pretty strong ones that can kind of clean up the leaf surfaces a bit.
I don't have much experience with fruit production and how you handle your fruit production operation, if it's UPIC or if it's for wholesale. So I think if you try to keep picking when it's dry would be a big help. And if you have U-pickers, they're probably not going to be coming with gloves, and you can have a foot bath where they probably wouldn't have a problem dipping their feet. But beyond that, I don't know.
So, like in the past 10 years, it's been FSMA, your workers can't wear plastic gloves picking berries because of the bacterial carrying of the plastic versus the human skin. So can a programmer say and use an oxidate type product on a daily basis somewhat sanitize that field? In other words, is that a protocol we could use, say, my crew's in there picking, we don't do UPIC, but that was one of the reasons we can't do UPIC and still meet FSMA needs, because you don't know what kids snotting on the blueberries or peeing in the bushes or whatever. So or whatever else the customer may bring in there to meet those FSMA protocols. We don't do UPIC, but say a crew's in there and we'll pick berries till noon. Can it daily or every other daily spray of an oxidate type product enhance this control of these, or is this like because oxidate kills like whatever touches and then it's gone. So is that,
Yeah. Yes. So the question being, can we use peroxide-based products? Oxidate, I think, is hydrogen peroxide with peracetic acid. Can we use those daily after the pick's done for the day and get control of killing some of these spores every day? In my experience with using hydrogen peroxide-based products frequently, you can, and they are relatively safe. I think with oxidate specifically, that peracetic acid that's in there would be the only concern. So you might want to stick to maybe just standard hydrogen peroxide and find a rate that works for you. But I would say it's relatively safe to use frequently, at least 4 or 5 times a week.
Does anybody have any other questions? Over here?
we've talked mostly about the nursery stage, but what if you get your plants from the nursery, you plant them in your production field, and you see the neopene collapse and whatever? Is it best to, like you were saying, it lasts in mature mother crowns up to 18 months or longer? Is it best to just nuke the field and give up and try to get it out? Do you pick out your year collapsing crop? And then how you were saying with nursery production, you want to rotate 4 years. So obviously you don't want to plant back to strawberries, but is it going to transfer to other crops? Say you put a caneberry crop into that field. So just maybe touch on if you have an infected production field, how should you go about using that field after it's had an infected cycle?
Okay, so the question was, you've received plants from a nursery, you've planted them out, and then you're starting to see collapse. What might be the best process to reduce or eliminate the problem in the immediate time frame, but also in the long term?
I would say if you start seeing a plant that collapses here and there, it's all about reducing inoculum, so just rogue that plant out.
Further than that, I would agree with Nora. I think you want to get the plant out as fast as you can. Testing your plants before you plant them can be take a really sufficient sample size as big as you're comfortable with and send it away to the proper lab, and that might help you avoid getting in that situation in the first place. But aside from that, it would be removing the plants and then starting a pretty heavy treatment right after.
Any comments on the long-term process?
Is there anything that you can treat the sap whites that are in the soil the next year, if we rotate it every other year? We treat the land now with something while we lay it out that would speed up, killing the disease in the land. Like if you just rotate every other year, and it's a sacrifice that's going to be living on the dead tissue that you just dug. What are some of the things we might use in soil applied or disc it up that would help? From carrying to the next crop 2 years later, a year later.
Yeah. So the question was, if you don't have time to have a long rotation, to three.
What is your recommendation for what time frame? 5 years? 3 years? 2 years?
I don't know if there is there a recommendation, Marcus, is anything in Florida that from a time? I mean, you guys don't have an opportunity in a lot of cases to rotate for a long time, but is there any concept on how long those spores last?
Yeah, it lasts for a while.
Sorry, I'm going to get Marcus up here. He's our next speaker, but has some history with you.
Yeah, no, the spores last for a while in the soil, especially on the crop debris and the crowns. So the recommendation that we are using in Florida is, as soon as you are done with the fields, remove the plastic and keep disking the fields as much as you can to help breaking down the disease and breaking down the tissue and to make it easier to decompose. And that's the recommendation that we are using right now.
If we add nitrogen, would that help it break down faster?
That we haven't tested.
Would it would?
It could, yeah. Okay.
Thanks, Marcus. Sorry to put you on the spot, but couldn't help myself. Another question? I saw another hand up somewhere around. Okay.
I was just gonna make a comment. We have a lot of growers that don't have the option to rotate, so they have to go back to the same field over and over. And our recommendation is the day you quit picking, get those plants out as fast as possible, get slasting up. And we recommend all of our growers to get a cover crop out. So on our personal farm, the day the plastic comes up, I've dissed it, I've planted a cover crop. We try to get a good cover crop stand. And make sure to fumigate if you can. There's a lot of, we see in the industry, a lot of small growers are not fumigating anymore, and that is very scary. If you quit doing that, you're definitely increasing your risk when you do that. So I would try as much as possible to do that. With that being said, I'd say the main thing is don't freak out, because we've seen a lot of growers on my personal farm. I've had deals on my fruit production, like 4 out of the last 6 years, and we've been able to successfully have a good crop every year, just by being diligent. If we see a plant that's got an issue, we get it out, try to stay on top of the spray program. As soon as our U-pickers are out of the field, I've got a sprayer sitting on the other side of the road. As soon as they're out, the last car leaves, we try to get a sprayer in and spray it. We don't open our U-pick before 9 o'clock every morning, because we want our plants to get as dry as possible before anybody gets out of that field. That's just some comments I have.
Thanks, Austin. Okay. All right, if there are no other questions, I want to thank Nora and Kyle for being up here and sharing their experiences and how they're planning to keep supplying as clean as plants as possible. So thank you very much, Nora and Kyle. I appreciate the Okay, our last speaker for this morning, I sort of introduced Marcus Marin from the University of Florida. Marcus has been working very closely with NEOP for several years now and has been involved in some of the studies down there. So Marcus is going to come up and tell us some of the findings from Florida, and I think they seem to be at the forefront of research, and they've got unfortunately, the most experience with NEOP.
So good morning, everyone. Thank you so much for being here, and thank you for the invitation. And it's a pleasure to be here today to talk about diagnosing and managing Neopistalothiopsis in Florida strawberry. So one thing that I like to say to everybody is, when in doubt, submit a sample to your closest plant diagnostic clinic, because the right diagnosis is the first step for your right recommendation and what you should be doing to controlling whatever is affecting your field. Neopistalothiopsis is a pathogen with many faces. It affects any plant parts like fruits, roots, petioles, and leaves. And it can mimic so many other diseases sometimes, like on fruits, it can resemble anthracnose, or if you look at the root necrosis on lesion on petioles, can also mimic anthracnose disease. So the right diagnosis is the first step going forward. Okay, and one thing to keep in mind too is, depending on the weather conditions, the symptoms and the way that disease expressed in your field might change. For example, in Florida, whenever we are having a wet season, we see more leaf spots and fruit rots. But whenever we have a drier season, not prolonged leaf wetness events, not much rain, we start seeing more of the crown rot issues. And the crown rot, sometimes you see the plant collapse, which is the typical symptoms of the crown rot. But a lot of the times you see like stunted plants, the leaves turn purple, and they're a little bit weaker, and whenever you cut and open the crown, you might see the vascular discoloration, like you can see on the picture, that orange coloration inside the crown, or sometimes you only see the outer membrane being affected by the pathogen. So there is some variations in there, but again, when in doubt, submit a sample to your closest plant diagnostic clinic. Neopistalothiopsis started in Florida in 2017. At first, the outbreaks, they were linked to specific nursery sources, and by that time, Florida Brilliance and Florida Sensation was the varieties that were mostly being used, and they are very susceptible to the disease. Fungicides were not much effective under those conditions and with those varieties. So since then, our group started to move into the Neopistalothiopsis research to try to figure out what is going on, especially because the Neopistalothiopsis or Pistalothiopsis-like pathogens, they were reported on strawberries for a while. The first report was actually in 1973 that it was reported outbreaks with fruit rots, and so it has been lingering around for a long time. But what is new on this specific pathogens that we are dealing with is they are more aggressive. So we're moving into genomic data to try to understand and uncover the genetic basis of those pathogens that we were looking at. And here we have a big group that is called Neopistalothiopsis rosei. Before was called as Neopistalothiopsis clavispora or Pistalothiopsis longiceptula. So there has been a lot of change on nomenclature, but nowadays Neopistalothiopsis rosei is the big group with more concern on strawberry. And on that group, there's so much variation and genetic diversity. So all the isolates, they are pathogenic, but what happened is there is a mixed population. Sometimes the isolates or specific groups, they are not aggressive on strawberry, and these new emerging lineages of Neopistalothiopsis, they are more aggressive on strawberry. So lineage 1 is the first lineage that we first identified in Florida in 2017. Lineage 2 was also identified in 2017, but in Mexico. And a couple years ago, that lineage 2 started to be widely spread into the US. So nowadays, we do have 2 types of aggressive strains in the US, which is lineage 1 and lineage 2 of Neopistalothiopsis rosei. And when I mean aggressive is, if you look on the pictures, we test our isolates on detached leaf and detached fruit assays to determine their aggressiveness. And whenever we are dealing with an aggressive isolates, we start seeing symptoms developing in 24 hours. And usually that symptoms, it's coupled with high incidence and high severity. As opposed to non-aggressive isolates, it takes more than 5 or 7 days to start developing symptoms, and their incidence and severity is much lower, as you can see on the pictures. So Neopistalothiopsis has been around on strawberry, but what is new are those aggressive phenotypes that some way they have a new genetic makeup that is making them more aggressive on strawberries. So because we do have that complex population, we move into designing molecular tools and how we can diagnose the pathogen on a proper way, especially if we are dealing with the aggressive strains, which is the major concern to the industry. So we must know what we have on the sample as soon as we can so growers can start taking actions, changing their operations, and going ahead of the disease. So we publish HRM assays that we are able to distinguish the lineage 1 from the rest of Neopistalothiopsis. Then later we collaborated with Gid Shinabel's group, and together we developed a restriction enzyme protocol that we can distinguish lineage 1 from the rest. But unfortunately, those methods, it can give you false negatives if you were dealing with lineage 2. So in that case, you really need to rely on the isolations, especially if you work on a diagnostic lab, because usually whenever we are dealing with aggressive strains, the consistent of the isolations, that means like how much the recovery rate from whenever you isolate a symptomatic tissue on culture plate, you are recovering that pathogen. If it's consistent, regardless if you might have a false positive, then it's likely you have lineage 2. We developed some new markers in our lab. We have a student working on that project, and hopefully that publication will be out soon. But in the meantime, if you need help or if you want to send samples to our lab to diagnose Neopistolochiopsis or distinguish the second lineage, you can contact me or Natalia, and then we'll be happy to help you out. So on the GCREC plant clinic, we have a lot of support from our Florida Strawberry Growers Associations. So all the samples that comes in from nurseries that Florida growers get plants from, or even local growers that are members of the associations, they don't have to pay for the samples because the association cover the cost for them. So that has been helping our industry, and it's also an incentive for the people to start bringing samples to us, so you can see and survey the fields and have a better surveillance what's going on out there. So all the samples that comes in, we do have molecular assays. All the samples that come in, we screen for all those major pathogens and potential threats that could be introduced to Florida. For example, Fusarium. We don't have much Fusarium in Florida, but that's something that we are already ahead and screening for that, and also Verticillium. So all the samples that come in, we screen for everything, and our molecular assays, we are able to give growers 24 hours after the sample submission. They already receive preliminary results with what is in there, so they can start taking some actions instead of waiting for the culture isolation. So that quick turnaround is something that has been very helpful for us to manage diseases down there in Florida. And lineage 2, like I mentioned, it was widespread a couple years ago for other states, and a lot of states are sending samples to my lab to diagnose Neopistolochiopsis. And here you can see, like, we have been detecting both lineage 1 and lineage 2 in several US states, so both lineages are out there. But besides diagnosis, here are some things that we learned while we are dealing with NEOP in Florida, is that we have to use as many tools as possible to balance the disease triangle. The disease triangle has 3 corners. That means host, pathogen, and environment. So you have to hit all the 3 corners in order to manage the disease. Sometimes, like Nora was mentioning, you might have a pathogen, we might have inoculum out there, you might have a susceptible host, but if the environment is not conducive for disease, even though your plants are infected with NEOP, you might have a good crop, okay? So the 3 corners are the 3 things that you have to think about it whenever we are dealing with NEOP. So from now on, I'm going to touch base on inoculum sources, survival, cultural control, cultivar resistance, and fungicide application and timing that we have been using in Florida. As inoculum source, plants were usually coming asymptomatic, and then whenever we were planting in the field, we have to turn on overhead irrigation for about 10 days for plant establishment, and that increase of wetness and the weather conditions, then we start seeing the disease develop on those asymptomatic transplants. So before, like I mentioned, was very linked to nursery sources, but that's not the case anymore. After the disease started to be introduced to the field, we learned that the disease established in the field, especially on the crop debris that are in there, and especially on the crowns that are very hard to decompose. We had a study that a former student were doing in our lab, and the crowns really hold the inoculum, like Nora was mentioned. We were able to evaluate those crowns up to 18 months, but after that, we ran out of samples and we were not able to really continue tracking. But that information is really helpful because that crop debris is really a bridge that connects your 2 seasons. And in Florida, we don't do much crop rotation. We don't have that availability of land to doing that, so that's a big concern. And on top of that, the weather in Florida is very unstable, depending of the year. For a few seasons ago, I would say 2 seasons or 3 seasons ago, the weather has been dry. So NEOP, it is in Florida. You can easily find, but because the weather has been dry, it has not been a major concern as far as right now. And this season is the same. But in Florida, we do have some tropical storms and hurricanes that can really blow out the pathogens. Here is a graph that you can see, like in October, we had just a few farms. On the graph, you can see like in the squares are nursery source, and in the circles are farms. So we had a storm in November, and you can see that after November, it really blows up in other commercial farms. Okay, so storms are really a way that can spread the pathogen and have that conducive environment on the neighboring farms. Alternative hosts, that's something that we were looking at. So we went to nurseries, and we also survey the wild vegetation around the strawberry fields. So that's me on a forklift sampling oak tree. So basically what we did, we went out there, we collect any type of symptoms and asymptomatic tissue from hundreds of hosts. We brought to the lab, and then we inoculate on the strawberries to see if those isolates we recover were pathogenic or not. We sequence them to see if they match the strawberry aggressive strains, and we were not really able to find a major host or a major reservoir of that inoculum. So what we found is like the strawberry aggressive strains of NEOP, it really likes the strawberry debris. Okay, there were some instances that, especially in North Carolina, we recover aggressive strains from rhododendron, but the recovery rate was very low, and we went back to that, and we were not even able to recover anymore. So that question remains, like, who comes first, the egg or the chicken? You know, you might have on the strawberry first, then jump to rhododendron, but we don't think that will be the other way around. So managing the crop debris is really key because we didn't see, especially in our areas in North Carolina, that wild vegetation will really hold that inoculum as a major source of inoculum. We also look at cover crops because in Florida, after a lot of growers, after the cropping, the strawberry season, they destroy their crops, then they plant a cover crops for a few months just to increase the organic matter and soil benefits that the cover crop gives to them. So we were looking at whether the cover crops will serve as a major host for NEOP, and you harm the disease management. And what we found out, and yes, NEOP can infect those cover crops under artificial inoculations because we use pure inoculum and we really force the disease to infect. But whenever we put all those cover crops in the fields, in several field trials where we had infected plants, and we know the fields were bad with NEOP, so we had plots, big plots with those cover crops. We monitored that over more than one season, and we didn't see any indication that the cover crop was actually being infected by NEOP at field conditions. So our recommendation, yes, for these cover crops that we tested, they can continue to be used, and they will not harm the NEOP management recommendation. And here is also a question that we get very often. Start with clean transplants is one of the best recommendations, but that's not the reality sometimes. Sometimes you get transplants that are infected. So a lot of the growers were asking about cultural control or removing the leaves to reduce the inoculum, how effective that is. And again, you have to keep in mind the disease triangle. If you don't have a conducive environment, you might go there, remove a few leaves, and then the plants start to grow nicely, or even if you don't remove leaves, you might have a good crop, but if the weather is conducive, that can change. And here is a trial that we had in our area. That was a very conducive year, so we had a lot of leaf removal from a few of the treatments. And the last line, if you can see, like we were removing leaves of all, like for example, if the leaf had one leaf spot, we were removing that leaf. We were controlling the disease, but at the same time, we were harming the yield because we were removing photosynthetic area of the plant, and we were really harming the yield. As opposed to if we had no leaf removal and the fungicides recommendations, you had the same level of disease and higher yield. So in that case, on that particular trial, the recommendation would be don't do leaf removal, just spray the fungicides and keep praying for good weather. Also, we have been moving into cultivar resistance and working closely with Vance Wintaker to develop cultivars that are more tolerant to NEOP. In the orange or on the yellow boxes, you can see the new Florida varieties that were released. That's Amber and Encore. Those varieties is a bit more tolerant than Sensation and Florida Brilliance, as you can see. And so we are moving to the right direction. It's going to take a bit of time to have like a complete resistance, but just having that tolerance is really helpful to our industry right now. And like Nora was mentioning, having cultivar trials to your local area and see what cultivars that are commercially available that are resistant to NEOP that also gives you a good yield is a very promising type of study that should be conducting in several areas. Here's, for example, we had a trial that we were looking at inoculum concentrations because we want to see that, okay, how much of inoculum, how much of NEOP we need to hammer the plant in order to have an outbreak. As you can see here, concentrations above 10,000 spores per ml, we do have an outbreak on both a highly susceptible variety, which is Sensation, and a moderate susceptible variety, in this case is Florida Pearl. But lower concentrations, less than 100 spores per ml, the plants looks fine. You might see some symptoms. We harvest them, we compare the yield data, and we don't have yield impact, even though NEOP is in there. So the take-home message from this trial is we don't have to completely eliminate NEOP out of the system, but if we use those tools to lower the inoculum level, we can really survive and have a crop and go through the process, okay? And here is another trial that is the same trial, but looking at the data in a little different way. Here we are looking at the impact of the weather coupled with the genetic resistance. So if you look on 23-24 season, on the gray bars, we are looking at Sensation, which is highly susceptible, and the black bars, we are looking at Pearl, which is moderate susceptible. And the data here is on incubation period. Incubation period means after our inoculation, how many days it took for us to see the first symptoms on the plants, okay? So the more days, the more time the disease had to develop. On 2023-24, we had some rain events, so the bars are shorted, especially on the concentrations above 10,000 spores per ml. But if we look on the 24-25, on the highly susceptible variety, it didn't change much from season to season. 24-25 was a dry season, but you can see that tiny bit of resistance or tolerance on Florida Pearl. If you look at 1,000 spores per ml, it took about 60 days for you to start seeing symptoms after the inoculation. So that tiny bit of resistance coupled with a good weather, it can take you to a long way. And also here on this trial, we were simply evaluating the inoculum concentration. We were not spraying any fungicides. So imagine if you use that tolerance, if you have a good weather and you use a good management recommendation with fungicides and sprays, that can give you a long way, regardless if you have NEOP or not. So that's a good light at the end of the tunnel. Okay, so as summary for fungicide application that we have been using in Florida, the strawberry pathology program, we have been using a lot of collaboration with the industry and partners on testing several different products. So Tarum and Switch are very effective on controlling the disease. So our recommendation is to use Tarum as a basis application, and then use Switch only whenever you have a rain event or prolonged leaf wetness, or whenever we have a highly conducive weather condition, okay? So some DMIs are also effective, like Guido already mentioned, so I'm not gonna get into details in that. For nurseries, the recommendation is to use the chlorothalonil, Tarum, and if you are able to use Omega, that's another product that's very effective. And we are asking nurseries to avoid using Switch because it can harm the fruit production later with disease resistance, especially for anthracnose, botrytis, and even Neopistalothiopsis, okay? So besides having the right fungicide, we also want to see, okay, can we reduce the fungicide application, and what is the right timing for fungicide application? So in the lab, we have studied the Neopistalothiopsis, and we learned how much wetness condition and what are the temperatures that the pathogen need to germinate, cause disease. So all those factors we're getting into account whenever we develop a model to forecast the disease and to really aid fungicide application in the field. And the idea for that is to put that data into the strawberry advisory system that's already in place for anthracnose and botrytis. So this year we are validating our models, and that's already our 4th season that we are using these models to validate, and they look very promising. If you see on this table here, if you use Tarum every week and Switch only whenever you have alerts, we got like 4 applications of Switch and 16 applications of Tarum. But if you use the models that we are developing, if you look at Tarum, you can reduce the number of applications to more than 50%. So that's very promising because you reduce the number of applications and you use the product only whenever you need in case of Neopistalothiopsis. And here we are testing susceptible varieties, and which is Sensation and Florida Pearl, and remembering that Florida Pearl is a bit more tolerant to the disease, and both of them are working very nicely with the model. So this season is our 4th season. We hope to soon be able to incorporate that system into the strawberry advisory system so they have alerts for Neopistalothiopsis. And here is a picture of our plots. And as you can see, we have the non-inoculated control, the inoculated control plots, and on the treatments where we use the models to aid fungicide application, you can see that plants looks nice, they look healthy, they might have a little bit of disease, but not much yield impact. So that's very promising, and we're very looking forward to that. So here, just a take-home message and a summary for what we have learned and what we have been doing with Neopistalothiopsis is avoid introducing infected plants whenever you can. Spread the risk if you have the option of planting. I know everybody likes to plant some of the susceptible varieties because they have some fruit quality that is desirable by the market or other features that you might like, but try to spread the risk as much as you can. Plant a little bit of a more tolerant variety or things that you have more information on. The long-term solution, it is the breeding like I mentioned, the breeding programs are really moving towards the right direction. So having the cultivars that are more tolerant and testing on your geographic area to see the performance of those varieties are really important moving forward with Neopistilotropsis. Taran, Switch, and DMIs are good to be used. Taran has a basis fungicide. Switch and DMIs whenever you are having a high alert or a conducive condition for the disease, and chlorothalonil for nurseries, okay? Remove and destroy all the crop residues as you can. I know if you have a small operation and if you have the capability of removing all that crop residue, that would be the best. But if you don't, just keep disking your area, destroy as much as you can and eliminate that crop debris. And fumigation is also widely used in Florida, and what we learned from our trials is that Telone C35 is more effective on controlling not only Neopistilotropsis but other fungal and soil-borne pathogens as well. So use fumigation as much as you can. I know it's expensive, but just see your areas, but in Florida we widely use that and helps reducing the inoculum that remains in the field. Okay, level the fields whenever possible to avoid standing water and improve the water drainage. That's something that we saw, especially in a few nurseries that we visit. Depending on the way that the water drain inside the field, you can really see that if you have an infected area on a higher elevation and then you have the water running through your field, you can easily see that the spores are really moved with the water and you can track them. So improving the soil drainage and leveling that field is very important. Wash equipment and sanitizer for workers whenever they are working on the field with quaternary ammonium. It's highly effective. That's something that we learned. We had trials that we had people harvesting infected areas. We were collecting the gloves and evaluating in the lab. And the amount of spores that can be attached on the gloves and also the clothes on your arms is very impressive. And whenever you finish harvesting, you touch the plant and you can really see that transmissibility, and we proved that in the lab. We have a student working on that. So if you have an infected area, try to work on that area at last. So move your operations. And if you have a crew harvesting or something, use the sanitizers in between the operations and clean as much as you can to avoid cross-contamination. They might not be the major source of inoculum, but every little bit that you use to lower inoculum and avoid cross-contamination, that helps on a long run. Okay, so tools and supplies, again, you can use quaternary ammonium to clean them. And one thing that we have been using, especially in our trials, and we have been testing, is using our plant sauna to use temperature to sterilize those equipment. And if you use 60 degrees for at least 1 hour, that's a good option that you can use to sanitize the equipment with temperature. Okay, so besides that, I want to acknowledge my lab and the strawberry pathology program and Dr. Natalia Perez. We work a lot together. And if you have any questions, just let me know. Thank you.
Do we have questions for Marcus? Any questions? Yes. You know.
Nora mentioned that she talked to somebody from...
The question was asking whether there was any work or understanding of any predisposing factors to NEOP that might affect how it impacts strawberry plants?
Yeah, we haven't looked at close to that perspective, but just by experience analyzing strawberry samples and the time points in our season, what we see is that it's linked to plant stress. So for example, we did had a trial that we were comparing, okay, like if you dip the roots on a spore suspension versus only putting spores on top of the soil pot just to mimic, okay, the different types of inoculations and how harsh one inoculation will be with the other type. And we saw that as more stress the plant goes through, especially the bare roots, it can be more susceptible to Neopistolochopsis.
So, Jayesh, can you repeat the question? I think what you were asking was, have you found any different spray materials or any different, strains, sorry. Any different strains over the last 12 months?
There, like I mentioned, the Neopistolochopsis rosei group, it's one of the groups of species that affects strawberries. There are so many others that are reported, like, for example, Neopistolochopsis iranensis, Mesopotamica, and a few other species that has been reported in other areas. But we do have those in Florida as well, but they are not aggressive like those two lineages that we are seeing more issues. And a lot of the times they are more associated with stressed plants, or you can co-isolate them with other pathogens like Phytophthora or Macrophomina or Coletotrichum. So there is no indication that those other might be the major pathogens. They coexist with some others, but not the major ones. So as far as of aggressive strains, we are only seeing those two lineages that are within Neopistolochopsis rosei.
Yes, go ahead, Victoria.
In a system where you don't have Thyram, like a Canadian system, I noticed you have chlorophyllol for your nurseries, but do you see fit with chlorophyllol in the growers system if Thyram?
The question was whether you could, in Canada, chlorothalonil is available for fruit production with some limitations, but would that, we don't have the availability of Thyram, but would chlorothalonil fill that void, or would be helpful?
Yeah, no, that's a good question. And that's a big concern. We are afraid that we are losing Thyram here too, and that would be a big loss for strawberries and also some other small fruits in general. Yeah, the problem is, that's a good question. You know, like, yeah, what we learn is like, I mean, like the recommendation that we have right now for Thyram and Switch is something that we have been like testing and proving how effective it is for a long term and several seasons. So probably something to still be tested. Okay.
Okay, if there aren't any more questions, I want to thank Marcus once again for a great presentation. I want to thank everyone for participating this morning, and I thank our sponsors, as always, for their continuing support.